A'udzubillahi minasyaitonirrajim. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Welcome back to the SBT 1322 Cell Biology. This will be the lecture for the week 12 with the topic of signal transduction mechanism. So this signal transduction mechanism is something that you have to know when we talk about the cell communication. The cell-cell communications involve the transmissions of signal, okay, between the cells so that they can communicate between each other. So this week we are going to talk about the components of the signal transduction mechanism and a few examples of signal transduction mechanism inside a cell. So here is the objective of the lecture, which at the end of the lecture you must be able to explain. The first is the chemical signal and cellular receptors. Second is the G protein link receptor. The third is the protein kinase associated receptor, and also number four is the hormonal signaling as the example of a signal transduction mechanism or cell signaling in cell biology. Let's recall your knowledge library. Do you know what the meaning of cell signaling? What are the components involved in the cell signaling? And what are the basic mechanisms involved in this cell signaling? So you know that the cells can communicate between each other, but how do they communicate? Because they don't have mouth, they don't have ears, they don't have nose, they don't have eyes. So they are purely depending on the signal that they can convert it into kind of information for the cells to react to that. The basic concept of cell signaling depends on these two properties. The first is the cell is active, and second, the cell is dynamic. When we talk about the cell is active, what does it mean? Active means it has a metabolic activity. So it's more on the metabolic activities. While for the dynamic property, it is depending on the ability of the cell to respond to the external environment. So these are the cellular responses, okay, against the uh, external environment. But how can this be achieved by a cell? As I did mention before, they don't have any other sense like us. We do have eyes, we do have mouth, we do have ears, we do have nose. But for the cell, they don't have that. Okay, so the basic concept is really important in this topic. So you have to look into the basic mechanism. How can these cells communicate between each other? And in most of the time, the cells communicating by using the chemical signal. There are four main cell signaling mechanisms in cell biology. The first is endocrine. In endocrine cell signaling system, they uses hormone as the mediator or as the signal to the effector cell. Second is the paracrine. Paracrine is much more local compared to the endocrine system. In paracrine, the mediator is the local mediator, so the target is only at the neighboring cells. For the neuronal, it is specific for the uh, neuron cells, and it's involved neurotransmitter to be used as a signal mediator. While the last one is the contact dependent, where it has the direct contact between cells and is mediated by the membrane-bound signal molecule. Okay, it is similar to the major histocompatibility complex (MHC), where the white cells or antigen-presenting cells know they are the self or non-self cells. There are few elements that involve in the cell signaling. So what are they? Based on this diagram, you can see that the first important element is the ligand. Okay, ligand is the chemical or compound outside the cells that will be recognized by the receptor, and this ligand we call them as primary messenger. This ligand only communicate to the cells by attached to the receptor on the surface of the cell. So that means the ligand remain outside the cells. So, and the second one is the receptor on the cell. Okay, so the first is the ligand and the second is the receptor. So the first event in the cell signaling is the receptor ligand binding. So the binding of this ligand on a specific receptor, which later will convert that interactions to the signal transduction. Okay, so the second component involved is the signal transduction, and this can be achieved via second messengers. There are various types of second messengers, and we will look into a few examples later in this session. And this second messenger will get to the effector component inside the cells for the cellular responses, or in most of the cases, this second messenger will activate the transcription factor in the nucleus so that it will increase the gene expression. Okay, it can be increased or decreasing the uh, gene expression depending on the type of signal or type of ligand that the cells receive from outside the cells. In the simple diagram over here, we have to repeat this again. First, the main component involved in the signal transduction is the ligand itself, which happens outside the cell. And the second is the receptor. They are various type of receptor, but in this topic, we are going to look into two main receptors, which is the G protein coupled receptor or GPCR, and the second one is the tyrosine kinase receptor or TKR. Okay, the third component is the transducer itself. Okay, the activations of the transducer will cause a series of activation inside the cytoplasm or the cytosol, and this will increase the number of second messenger. This second messenger will give an effect to the target protein, which later will cause the changes in the metabolic activity or the gene expression if it is about the transcription factor. So I want you to understand about what are the definitions of ligand, what are the definitions of receptor, transducer, effector, second messenger, and also target protein or DNA. And you must be able to outline few examples of each of these uh, component, which we will go through in the next slide. This diagram shows the general pathway involved in the signal transduction mechanism. The signal in the form of hormone or compound from the environment outside the cells will communicate to the cells by attaching to the specific receptor. And this receptor uh, will relay a series of proteins and second messengers until it reach to the activations or uh, alterations in the metabolism. So the three main steps involved over here. First, the reception. Second is the transduction, and third is the response itself. The reception involves this part, the ligand and receptor. The transduction is the conversions of receptor and also amplifications of signal by the second messenger until it reaches to the effector. The effector will give a response and it will cause the effect that we can see on the cell. So this is a basic pathway, and you must remember that the ligand is the compound or chemicals that remain outside the cells. So the ligand only shaking their hand or convey the message to the receptor. And then the receptor is the one who actually tell the transducer to give second message. Okay, they convert the message into a message that can be understand can be understood by the uh, effector cells. Okay, the effector component inside this cell. Over here, we can see that the signal and the transduction mechanism. Why we need the transduction mechanism inside the cell? You may think that one signal will cause one reaction inside a cell. Actually, it's not in the reality. Usually, the effect inside the cell is um, amplified versions of the signal. So let's see the example over here. We do have the X molecules of ligand, and this X molecule of ligand will attach to the X molecule of receptor, and this receptor will convey the message or translate the message to the transducer to give 20X of molecule. And this molecule will activate this series of activation involve a different type of enzyme inside the cell. And if you can see here, it goes to 10 times, 100 times, and also 1000 times, even up to 10,000 times effect, even with the signal or one-time molecule. So that's mean the series of amplification, okay, amplification involved in the signal transduction. If one signal, one reaction, then we need more ligand, which will cause a toxic effect to the cell themselves. So this is how we actually control the human body control what can we process from the external signal and make it into uh, effect at the cellular level, which in a cumulative way it will cause an effect in the tissues or organ level. The next section is about the chemical signal and cellular receptors. All right, in this section, you will learn about the ligand and receptor involved in this signal transduction mechanism. So, uh, so the basic things that you must understand is first, what are the chemical signals that can trigger the signal transduction, and what are the cellular receptors available on the surface of cells that can actually lead to the transduction of signal by secondary messenger. The main question that you have to ask yourself is what kind of environmental factors do cells respond to? For example, some cells can respond to the level of glucose. Some cells respond to the level of insulin. Some cells respond to the concentrations of oxygen or carbon dioxide. So how can these environmental factors be represented as a ligand or signal in themselves to make the cells to respond to it? So this is where you learn about that chemical signals and also the cellular receptors. So that means the signal must suit to the different type of receptors so that the cells can actually recognize them and will able to respond to it. In other way, the chemical signals, we do have two types. Remember about the endocrine system and paracrine system. So what are the difference between these two signals? The hormones or paracrine mediators? If you look at here, we can see that the major differences between the hormonal system and also the local mediator. When we talk about hormone, they are much more systemic because the hormone was secreted into the bloodstream and will be carried out throughout the body by the systemic blood circulation. And along the way, the mediators will trigger different type of cells that may have the the receptors specifically for that hormone, and that receptors or that target organ can be local or can be away from the site of signal initiation. So in other way, we can say that the hormone in endocrine system, the hormones actually are the systemic distribution, while the local mediators, it is the signal between the cells that trigger and also uh, excrete those ligands towards the neighboring cells. While this one it is more systemic. So in other words, the ligands can be hormones or can be a local mediator. And you must know that between these two, they can be categorized into two types of polarity. First is a polar ligands such as insulin, and the other one is the non-polar or steroidal hormone. The differences between the polar and non-polar is how do they actually attack or give the effect to the cells. For the polar, for example, insulin over here, they can only interact to the cells at the receptor on the surface of the cells. While for the non-polar, just like steroidal hormone over here, it can actually go straight to the cytosol and then go to the uh, nucleus and activates the expression of a transcription factor in there directly to the nucleus. So it has a measure that we can say that okay, this ligand it is specific and it also has certain degrees of affinity towards certain receptors. This is where we can see the functions of those ligand in the form of agonist or antagonist. So this is something that you have to search yourself. What are the definitions of agonist and what are the definitions of antagonist? So some of the example of extracellular signals that uh, usually been used as an example in textbook written over here, and you can get it from this reference. Another question that we have to ask ourselves is about the ligand is about the attachment between ligand and receptor. What kind of attachment do they have? How about the affinity? What are the means of this affinity and how can we measure that? In a simple word, the receptor affinity is the relationship between the concentrations of ligand in solution and the number of receptor occupied. So this association and dissociations of affinity of this ligand can be expressed in the numbers, okay, by using the constant of dissociation, or we call it Kd. If the receptor have low affinity, it will show that it has a high number of Kd. That means the 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 dissociation is higher than the high affinity receptor. Another additional question over here is about the mean of receptor downregulation. Okay, cell can change to sense change in ligand concentration rather than the fixed ligand concentration. When a ligand is present and the receptor are occupied for a prolonged period of time, the cells adapts or become desensitized, so no longer respond to the ligand. And this will cause the receptor downregulation, which is the removal of the receptor from the cell surface, which the receptor-mediated endocytosis, for example, and alteration to the receptor that lower its affinity for ligand, and these alterations to the receptors that render it unable to initiate changes in the cellular function. So that means the downregulations can be in terms of the sensitivity or affinity of the receptor, or the population, the number of populations of that receptors on the surface of a certain cell. And this is why we need to understand why and how this receptor uh, play its roles in the signal transduction. Because without this, we won't be able to understand what are the differences between the diabetes type 1, diabetes type 2. And even diabetes type 2, is it because of the insulin intolerance, or they don't have the ability to recognize the insulin? One of the popular example of the receptor is the G protein links receptor. Okay, you have to understand that there are two main type of receptors associated with this signal transduction. The first one is the protein link receptor, just like this G protein link receptor, or GPCR. And the second type is the enzyme link receptor, which you will learn about the tyrosine kinase, uh, where the phosphorylations of amino acids by the use of ATP as the source of the phosphate, and this will lead to the activations of the second messenger. Let's have a look at the definitions of this G protein. So what is a G protein? You have to know the structure and the functions of this G protein, and what does this type of receptors looks like. Okay, over here we can see that the G protein link receptor, it has 1, 2, 3, 4, 5, 6, 7. That's why they are also known as the 7 transmembrane receptor. It does have the uh, N terminal at the extracellular surface, and they do have the C terminal in the cytosol. They have 1, 2, 3 extracellular loop, and 1, 2, 3 not intercellular, it is intracellular or cytosolic loops. The binding site occurs at this loop at the extracellular surface, while the one that interacts with the G protein is somewhere inside the cytosol. This means that the ligand will come and bind to this receptor, which will later will cause a series of activations over here. The changes in the configurations, or 3D configurations of this receptor will cause the G proteins dissociated and being activated. This is another image of 3D configurations of the 7-membrane receptor, or the GPCR. And we can use this beta adrenergic receptor as an example of the GPCR. And the things that can actually bound to it is the epinephrine and norepinephrine. So the G couple, how does it works? The ligand binding cause the G protein to associate with a receptor, and this, for example, okay, the ligand will bind to here, and it will cause this alpha, beta, and gamma subunit of the G protein will dissociate, and this is because of the changes in the configurations of this area, and the binding causes GDP release. So over here, then they will have the ATP, and it will be converted to the CMP by the adenylate cyclase. So here is the example of the activations of G protein. Let's have a look over here. First, the ligand bind to the receptor, okay, and it will cause the GDP to be released, and the GTP will bind to the alpha subunit. Okay, it will be like this, alpha subunit. So the complex of uh, alpha, beta, gamma of G protein were dissociated, and it will cause the alpha releases GDP and binds GTP, and also the activating the G protein by separating the alpha domain or alpha subunit of the G protein from the beta and gamma G protein. So the subunit separates, and this alpha subunit of G protein activates or inhibits the target protein, while this other subunit remains there, and this alpha subunit hydrolyzes its bound GTP to GDP, and it will become inactive and will form a complex with the beta and gamma subunit again, which the complex will return to the intracellular loop of the GPCR, and this is what we call a subunits recombine to form inactive G protein. So the cycle will start back, okay, when the ligands bind to the receptor. Here is another diagram showing that how this G protein complex, okay, which have the alpha, beta, and gamma uh, form a complex when they are inactive. Okay, when the ligands come in, okay, when the ligands bind to the receptor binding protein, so this G protein complex will release the GDP and taking up the GTP to the alpha subunit. This will lead to the dissociation of alpha subunit from the beta and gamma subunit, and both of these subunits can be active. Okay, so during the activations, it will hydrolyze the phosphate to become GDP back, and it will remove one phosphate to get the GDP, okay, GDP, and it will become the inactive G protein again, and will be coupled to the G protein link receptor. So what are the impact of the activations of G protein? The G protein, the alpha subunit, will couple to the adenylate cyclase, or we can call it as an effector, to convert the ATP to become CAMP. And this CAMP is known as a second messenger, and it involved in the series of amplification, as we can see from the third slides earlier. Here is the example of the second messenger. How this second messenger actually been produced by the protein kinase A. So over here, CMP act as a second messenger to activate the protein kinase A. Remember, in the previous slide, I did mention about the adenylate cyclase will convert the ATP to become CMP, and that will be the effector from the activations of G protein. So now the CMP will act as a second messenger to activate another enzyme, which is the protein kinase A, and this is considered as an allosteric activator. The protein kinase A, it has the regulatory and catalytic subunit, which means if the CAMP bound to this regulatory protein, it will cause the dissociations of regulatory subunits from the catalytic subunit, and it will cause the activations of catalytic subunit, okay, like this. So if the CMP release from the regulatory, then this catalytic subunit will come back to the regulatory subunit. The example over here is through this phosphorylation, it will cause the transcription factors to increase its expression factor, and its later will cause the increase in the cell growth. As I did mention before in the previous slide, the protein tyrosine kinase is a phosphorylizing enzyme. Okay, so that means it induce the phosphorylation. So what are the phosphorylation? Okay, phosphorylation is a common activation or the activation strategy in the cell signaling, and this changes protein conformation of the protein. That means the 3D configurations drastically, because the hinge at the phosphate between the ADP and ATP is somehow very different, and it has different rotations between them. And as we can see here, it involved the covalent modification and the middle range time effect, which is not too fast and not too quick. The example of the phosphorylation enzyme is the protein kinase A. Protein kinase A modulates the activity of the enzymes that carry out work through the phosphorylation. So this is the keywords. For example, the adrenaline binding leads to the pKa, activating the enzymes that release glucose from storage to be used. And this is how it works. So the ligands come to the receptor, and then the transduction occurs, will cause the activations, and this will cause the phosphorylation and get to the target protein, which will in the end will get to the either changing in the metabolic rate or activities, as well as the gene expression. All right, for the turning off pathway, just additional question that you have to ask yourself: can this activation and these activations of signal signaling to be turned off? All right. So what would happen to the receptors when these turn off, and what happened to the G protein? How about the second messenger and about the phosphorylated enzyme? Actually, in the ideal conditions, some of the activation and inactivations are in balance, so that the production is in balance, and the net difference is zero. However, when the turning off of all molecule, it will cause to the or the detrimental effect, which is the apoptosis. This is the example of the how can this G proteins works in the many pathway. As I mentioned before, the G proteins in the previous example is the alpha subunit activating the adenylate cyclase, but the G protein can also activate this phosphorylated pathway, and this is the example of receptor which give a crosstalk to the phosphorylated pathway. All right, so I want you to read this, and we can discuss it during the synchronization or during the Q&A session. And this is how it looks. Okay, if we map the network of activation, inactivation, crosstalk between the second mediators, even it's come from the same uh, receptor, okay, and you can see that they actually giving the balance between activation and activations, and even the same messenger, it can actually give a different effect to the different effector inside the cell. All right. Okay, so this is the example of the agonist promoted signaling and agonist inhibited signaling. So this is how it's promote the 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 positive effect, and this is how it's actually uh, cause the opposite way compared to this agonist promoted signaling. All right, so for the calcium ion, this will be discussed during the Q&A session, and I hope that you can look at the importance of this calcium ion as the second messenger in organelle or in a membrane bound organelle, inshallah. There are a few questions as well on this topic about the calcium ions, but how do we know that the calcium ion involved in cell signaling mechanism, which you need to do some reading, and this is what actually happens for the uh, mechanism that involve the calcium ions in their signaling, especially to the concept of muscle contraction, where we need the calcium ion for them to actually give, in fact, to the muscle cells to actually either contract or relax. And another example is the use of calcium ion in the development of animal, especially during the fertilizations of ova by sperm. This will actually causing the calcium ion channel to be activated, and it will causing the influx of calcium ion, and that's why if you really look at the electrochemical properties of the fertilized egg, they actually will cause a very quick and rapid spark, okay, spark formation due to the sudden change in the calcium ion concentrations between the cytosol and the extracellular environment. Okay, as I did mention, the calcium ion involved in the promoting the relaxations or contractions of smooth muscle, as well as the endothelial cells for the vasodilatations and vasoconstrictions. All right, so the second example is the protein kinase associated receptors. Okay, so the first part we to learn about the G protein link receptor, and here is about the protein kinase associated receptor. That means there is no more G protein involved in this receptors concept. The In the protein kinase associated receptor mechanism, the signal will actually attach to the receptor, and then the phosphorylations of the tyrosines in the cytosol domain will cause a series of activations of certain other enzymes, which leads to the effector component to give an effect. Let's have a look at the protein kinase associated receptors. Okay, so what do all these receptors have in common? Okay, the tyrosine kinase receptor, they have the binding sites, the outside the cell, the extracellular environment. They have one adapter in the transmembrane region. They have the tyrosine kinase in the cytosol, and they have the tyrosine cytosolic tail. And these are the region where the phosphorylations will cause, okay, the phosphorylations of uh, ATP, okay, to become ADP, which means the tyrosine kinase, okay, when we talk about the kinase, it has the ability to make use of phosphates and bring it to other molecules. So it actually transfer the phosphate from the ATP to other molecule, and the concept of phosphorylations of a secondary messenger in there. All right. In the complex way, we can see that the example over here, the EGF, the epithelial growth factor. Okay, the EGF will be recognized by the EGFR, which means the epithelial growth factor receptor, and the attachment of this ligand will cause both dimer of this tyrosine kinase receptor come close together and become the active dimer. All right, and this will actually cause the kinase to make use of GTP and convert it to GDP. So this phosphate will be used to phosphorylize the tyrosine. Okay, the example over here, one dimers of tyrosine kinase receptor need 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 phosphate for them to phosphorylize the tyrosine at the cytosolic end of the tyrosine kinase receptor. And this will actually transduce the protein, okay, which will convert the PLC gamma to become this PLC and make it active to convert this PIP2, and this PIP2 will actually cause activations of not activation, actually the PLC phospholipase C gamma will digest to the to cut over here and leaving the DAG part, okay, the acetylglycerol part, and releasing the PIP2, and they add one phosphate over here to become uh, inositol triphosphate, and this inositol triphosphate will actually uh, in contact with the calcium channel to cause the efflux of calcium ion from the endoplasmic reticulum, and this will actually cause the increase in the calcium ion inside the cytosol, and this is what happened during the muscle contraction of the muscle cell. Another way of the cell signaling is through the growth factor, just like I did mention before, is EGF, epithelial growth factor, and it involves the involvement of Okay, so the example of growth factor and the target cells, and also the type of receptor complex involved in this signal transduction caused by the growth factor as the ligand. All right, so you don't need to memorize this. What I want you to look at is uh, what are the natures of this growth factor, and what kind of targets that it has the effect of, and relate this all together with the type of receptor complex that react or recognize those ligand. And this is what we did mention before, where the EGF uh, or the example over here is the fibroblast growth factor F, GF, and do they have the same signaling transduction mechanism as the EGF? All right, so same goes to this TGF beta, and uh, as well as the other growth factor. So what do these different pathways have in common? That's the question that I want you to look at. And what way is cancer related to the growth factor? What makes this growth factor become a detrimental effect and causing cancer to the cells? All right, so there's a relationship between the, remember when I talk about the balance between the positive effect and negative effect of a certain signal, and they have to cross talk between each other to coordinate the balance of cell cycle. Okay, and the next slide is about the hormonal signaling and how these endocrine and paracrine hormone differentiate themselves. Okay, you just repeat the introductory parts that we had before. Uh, so between the paracrine and hormone growth factors, so what are the difference between them? Right? And what are the some physiological function controlled by the hormone? Okay, so these are the things that you can actually answer that by looking at the table in the previous slide. Right, so I just repeat this again. Uh, and yeah, you do not need to memorize this, but you have to understand the mechanism, the basic concept of mechanism. All right, so the adrenergic hormones and also cholinergic hormones, how do they works, and how they actually interact to the receptor, and what are the receptors needed for this kind of hormones. All right, so if you look in all other mechanism, you can see that if they are working with the G protein link receptor, they have almost the similar pathway, because it involves the adenylyl cyclase and things like that. So the important parts in the cell signaling is the specificity and also the affinity of the receptor themselves. So that's mean they have different site for ligand recognitions on the receptor binding, on the receptor side, which will actually react to the ligand, and the transduction mechanism will utilize almost a similar pattern of mechanism. If they are working with the G link protein, then the activation of G protein will actually cause the phosphorylations of certain second messenger, just like CAMP, and you can see here is the CAMP involved there. But if they are working with the tyrosine kinase, and it involved in the calcium ion channel and phosphoinositol uh, triphosphate mechanism, or biphosphate mechanism. All right, so the rest is just a question that you have to ask yourself, and will be your additional reading in uh, this subject. All right, so I think I just browsed this and hope you can read and understand the slide, inshallah. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.